Hello, everyone. I'm very glad to introduce the paper submitted to iMeta. The title is A Panoramic View of the Virosphere in 3 Wastewater Treatment Plants by Integrating Viral-like Particle-Concentrated and Traditional Non-Concentrated Metagenomic Approaches. Environmental viromics is an emerging hot research topic in global environmental microbiome studies. Municipal and agricultural breeding wastewater is characterized by high chemical oxygen demand. This eutrophic environment is a rich reservoir of diverse microbes. Numerous studies have extensively explored the diversity and genomic attributes of bacteria and archaea in wastewater treatment plants. However, our understanding of viruses in wastewater treatment plants remains limited. Especially, the abundant bacteriophages in wastewater treatment systems affect the operation of biotreatment systems and might be closely associated with the horizontal transfer of antibiotic resistance genes. When investigating viromes via metagenomics, researchers typically employ either viral-like particle-concentrated or non-concentrated sample preparation methods. Gaps remain in our understanding of the virome acquired through different sample preparation methods. This study aimed to compare the VPC and NC methods in virome exploration, characterize the panoramic of the viral communities in 3 diverse wastewater treatment plants, and uncover the active roles of viruses in wastewater treatment systems using metatranscriptomics. This study collected 23 samples of wastewater or sludge from each treatment unit of 3 WWTPs. These 3 WWTPs treated wastewater from duck farms, pig farms, and municipal sources, respectively. VPC and NC approaches were applied to process these samples, and viromic analysis was conducted. Metatranscriptomic sequencing and analysis were performed on the raw wastewater and activated sludge samples. This study identified 11,045 and 38,438 viral contigs from VPC and NC data, respectively, and clustered them into 35,647 representative viral sequences. The viral sequences obtained by VPC method were of high quality, dominated by eukaryotics in the phylum of Cressdnaviricota. The NC method yielded a large number and high diversity of viral sequences, primarily dominated by the bacteriophage phylum Uroviricota. The NC method is more suitable for studying bacteriophages in wastewater. The VPC method is more efficient for investigating free-living eukaryotic viruses, especially those vertebrate viruses that infect humans or animals. Wastewater treatment systems could not completely remove vertebrate viruses. Their relative abundance in the effluent was still high, posing potential health risks. The cellular fraction was mainly composed of double-stranded DNA viruses, with about 56.1% being lytic bacteriophages, while the free virus particle fraction was dominated by single-stranded DNA viruses, with about 87.7% being lytic bacteriophages. This study identified a total of 5,341 potential virus host associations. On average, each virus can infect 1.1 prokaryotic hosts, and each prokaryote can be infected by about 3.9 viruses. Moreover, the correlation between virus diversity and bacterial diversity was stronger in the NC data than in the VPC data. In the NC and VPC metagenomes, 827 and 99 AMGs were predicted, respectively. The NC metagenome involved more metabolic pathways than the VPC metagenome regarding AMGs. Therefore, the NC method is more effective in mining auxiliary metabolic genes. At the RNA level, various AMGs by bacteriophages were actively expressed and played an important role in the carbon, sulfur, and phosphorus cycles. In the NC data alone, 29 ARGs carried by bacteriophages were found, with Uroviricota phages being the primary carriers. The lincomycin resistance gene LNU-D was shared by multiple bacteria and viruses. Although viruses carrying ARGs accounted for less than 0.08%, the risk of horizontal gene transfer mediated by phage transduction cannot be ignored. This study systematically and quantitatively compared traditional metagenomics and virus particle-enriched viromics for characterizing the viral composition in complex environmental samples from multiple dimensions. The main conclusion is that the combined application of these 2 methods can characterize the environmental virome more comprehensively and accurately. If economic costs are considered, one of the methods can be chosen based on the research subject and objectives. Additionally, metatranscriptomic analysis at the RNA level revealed the activity of viral AMGs. The risk of viruses mediating the horizontal transfer of ARGs was also explored.